All right, welcome everybody. My name is Damon Tighe. I'm Bio-Rad's Curriculum and Training Specialist for the Western region of the United States. I'm going to be taking you through this webinar today on buzzing for bumblebees, basically their conservation, identification, and how DNA barcoding is basically facilitating both of those first two things, the conservation and identification. So there we go. All right. Seeing species is key to conserving species and understanding ecosystems. And we've been pretty good at this for a long time, but mainly using morphology to be able to kind of see species. So we collect species, we do everything like count hairs on their hind legs and things like that, and then look at the way that they, emerge from the winter, say for some of these bees. Are they coming in as one group? Do they come in different kind of positions throughout the season progression in order to kind of understand different species? And if we think about the number of species that humans have described on the planet, as far as large stuff, about 86% of species are probably still undescribed. I'm not sorry, not large stuff, but everything is pretty much undescribed. And that's mainly because a lot of this is microbial stuff that we're still just getting the toolkits together to be able to quote unquote see them. And a lot of these tools tend to be more molecular, because if you've spent much time with microbes underneath the microscope, you realize there's only so many shapes you can get that are about 5 microns in length. And that makes it really hard for us to identify them without anything except for molecular tools. So that's why in part of this talk, we'll be kind of highlighting how some of these molecular tools are helping us understand even large species like bees. So if we look at this slide here, what do all of these crops have in common? Well, all of these are bee-pollinated crops. And I know most of you are like, well, duh, everything's bee pollinated. Well, actually, quite a few of our crops out there, and most people don't think about this, are actually fly pollinated. So anybody that enjoys chocolate, you can only enjoy chocolate because we have flies that do the pollination and a number of other things. But bees are a really essential portion of a lot of the crops that we grow at kind of large scale, especially in the Northern Hemisphere. So we rely upon them, and that's why we see things like honeybee hives brought to croplands in order to increase pollination at sites. In fact, where I live in California right now, we have the giant almond bloom going on in the Central Valley. So there's tons of bee boxes out everywhere right now around those almonds, trying to make sure they get the highest pollination rate possible. So if usually what I do is I used to have students go through and pick what their favorite thing here on the slide deck was, and I used to bias this so that it was like really there's only one good treat, and hopefully it would be the blueberry. But I've had years where kids were like, the majority of the class picked peppers, so now I just kind of go, let's talk about blueberries. Blueberries are really important pieces of ecosystems. Organisms at every different scale eat them, so everything from your squirrel to your cedar waxwing to your black bears. And so beyond just being an important food crop, it's also an important piece of our ecosystems in the northern latitudes. In fact, there's such a key piece that it actually kind of upends some of the way we think about large megafauna in North America. So I know when people look at grizzly bears and they see them kind of bulk up for the fall, everyone's like, oh man, they must just be eating so many fish and eating so much meat. Well, a lot of the grizzly bears, especially in interior Alaska, are not bulking up on a ton of fish. They're bulking up on a ton of berries. So there was a grad student, man, and I'm glad I'm not this grad student, about a decade and a half ago, that they were basically tasked with figuring out how many blueberries these grizzly bears in Alaska were eating. So they were basically following a family around, collecting the turds, and then going through and counting the blueberry skins. And about 30,000 blueberries are consumed per day, June to September, for a lot of these internal to Alaska grizzlies. So that's a huge amount of mass, both on the berry side and ultimately on the animal side. So how you get that many berries produced, we've got to have really, really robust pollination. And this is something people, at least in the Northeast, have been very concerned with for a long period of time, because there's a lot of agricultural activity around the production of blueberries. In fact, so much so that you see states spending money to understand who's pollinating their blueberries. So in Rhode Island, they've got 41 species of bees that pollinate the blueberries that have been studied. Why is this diversity really, really important? Well, bees, just like other groups of workers, show up to work at different times sometimes. And so if you've got a group that only comes out when it's really warm, you might not be getting like efficient pollination of your crop. But if you've got ones that are also coming out in the cold, you get this kind of thing that balances out, right? So that you're getting a robust pollination of your crops. And as you can see here with just a few that have been highlighted, it's a really big diversity in what these bees look like too. But you'll notice that a lot of these tend to be these kind of larger bees. And this is important because a lot of these Vacinum genus, the genus that blueberries is in, respond really well to buzz pollination. And we'll get into that in a little bit. Kind of globally, and especially in the United States, there's been a lot of PR around the honeybee decline. And this is really just the tip of the iceberg for bees, but also just for insects in general, right? We're going through more or less an insect apocalypse, which, not to sound gloom and doom, but that's not good for any of us, because I sure as heck don't want to be out there hand pollinating all of our crops. And we're already seeing this take place in certain areas of the world where people are going out and trying to hand pollinate as the number of pollinators are decreasing. Bee diversity worldwide is seeing a huge decline, and this is really, really scary for our crops. You got to remember, honeybees do a fair amount of pollination, but they definitely don't do near all of it for the crops that humans eat. And so seeing a decline worldwide in bees is very, very dangerous in a lot of ways. There's about 20,000 species of bees worldwide that we are aware of, but there's a fair number that are still undescribed, and there's probably a number of them that are cryptic, where we think we know the number of bees in an area, but if we were to go in and look a little closer, we'd find that there's actually multiple species hiding under a single name. Bumblebees are a really interesting group within the bees, mainly because they are this much larger bee. They get up and get going in colder weather, and that's why we see them emerging all over North America right now. So they'll be out there way before the honeybees could be active and getting things pollinated. Their large size and their wing vibration makes them really, really good pollinators for a handful of crops that need buzz pollination. In fact, they are 6 times more effective at pollinating blueberries than honeybees are, and it's predominantly due to that size and that buzz pollination. Ecological importance, they're major pollinators for a diversity of plants, and especially a lot of these early blooming spring plants rely heavily on bumblebees for pollination. From a conservation point of view, there are major declines going on right now. So 89% decline in the American bumblebee, which is Bombus pennsylvanica, which basically more or less exists kind of east of the Rocky Mountains. There's at least 260 species in the United States, which means that identification can be a little bit challenging and overwhelming if it's your kind of first foray into looking at bumblebees. The National Forest Service, though, produces really, really nice free guides online for identifying bumblebees by your region. So they've got ones for the Western United States, Eastern United States, and then I think there's even one I've seen for Alaska that make it easier to do the identification. So it's been a while since you've thought about bumblebees versus a honeybee. Here's just a quick visual. Bumblebee, way bigger, way fuzzier, a lot rounder. And like I said before, usually up and going much earlier in the season than the smaller honeybee. And remember, honeybees in North America are non-native. So it kind of drives me a little crazy when I hear people like save the bees, because it's more or less the same as like saying save the cows. They're useful tools, but they're not necessarily the native thing that was here pollinating the diversity of stuff. So the reason bumblebees are such good pollinators for specific plants is this buzz pollination. And this is something that has been known about for over 100 years, but we didn't really get super good studies on this until about 20, 30 years ago. And it's basically that bumblebees and a couple of these other really large native bees, like carpenter bees, are really good at this. What they'll do is they'll land on a flower and they will start changing their wing beating pattern. In fact, you can hear this if you've ever watched bumblebees land on, say, like tomatoes or anything like a blueberry. You'll notice that after they land, all of a sudden you'll hear a change in the wing beat. And what's going on is they're increasing their frequency of vibration using the wings. And what it does is it actually unlocks pollen grains from the stamens. And so a lot of plants have basically co-evolved with bumblebees to have them be the predominant ones who are basically unlocking their pollen grains, getting showered in them, and then doing basically pollination. And this is really interesting because this means that some plants, are very, very much so specialists for bumblebees or a specific species of bumblebees. And so if you see that bee go away, all of a sudden you've got the danger of that plant not making it to further generations either. Like I said, bumblebee diversity in the United States can be complicated because we've got 250 plus species. So if you're really trying to get into this for the first time, one of the easiest ways to do this is to actually look to a place that's a smaller landmass and is really into flying stuff, which would be the UK. So the UK has a great series of talks online about how to identify, I think it's like there are 7 bumblebees, which makes it a lot easier to kind of wrap your brain around. And then maybe once you've done that, you can then kind of graduate and grab one of those National Forest Service documents and then kind of go through our diversity here. But the big thing to be aware of is the way that most people go through the classification of bumblebees is by looking at the different banding pattern of hair on the back. What color is it? How many bands are there? And things like that tend to be the main drivers of the identification keys. And this is just going to be important because in a little bit here, we're going to kind of play a game about identifying bumblebees in Colorado. So round 1 is we would learn to identify one bumblebee. And I know you're like, well, that seems easy. It could actually be tricky. Round 2 is you'd learn to sort kind of 2 bumblebees that live in the same habitat visually. And then round 3 will bring in a new tool. You can probably guess what that is, a molecular tool to sort bumblebees that also live in that same habitat. So the reason we're going to look at Colorado is because what I'm going to bring you through is actually a true case study that occurred a couple of years ago. And this is a game that you can play with your students. So we have resources online where you can print out these documents that students basically would get a set of cards to look at that are photographs of bees. And then they would basically choose whether or not this is the bee I'm looking at, this is not the bee I'm looking at. So it's like little baby steps into doing field taxonomy. So we'll move on to the first organism, which is Bombus huntii. So Bombus huntii is a bumblebee that we find all throughout Colorado, except for those counties as soon as you leave the Rockies. Huntii doesn't really make its way to the eastern United States. And then what we also see is it has an elevation preference. So it's usually between 4,000 and 9,000 feet. So all this data right here, I'm pulling just off of the National Forest Service identification cards for bees. I think there was actually a specific card just for Colorado where I could go through and look at them. So if I'm trying to decide if this is huntii, what I'm going to be looking at is basically these banding patterns. And so each student group would get a set of Bombus huntii to look at. And I feel like there's a slide here missing. Yes, unfortunately, there's a slide here missing where I have actually the identification banding pattern for huntii. So just play with me. We'll put it this way. Usually huntii, the way that it looks is this. You've got a black butt band, a yellow band, and then 2 orange bands, followed by another 2 yellows, and then a black stripe where the wings come across, and then more or less a yellow head is the easiest way to identify huntii. And what I've done is I've made 8 sets of these kind of cards for student stations. And so they would have these to look at. And these are all images that are pulled from iNaturalist. And if you're not familiar with iNaturalist, this is just an online platform and app where you can use your cell phone to record species biogeography. And in the process, it helps you kind of like learn what's out there. And so these are all images that I was able to go on to iNaturalist, go into Colorado for these counties and be like, give me some images of Bombus huntii, and I could kind of take them. And the beauty about pulling it from iNaturalist is I am getting it from the perspective of somebody in the field. And so sometimes you get a great angle and you can see that organism really clearly. And other times, maybe the angle's not great. And so you're trying to make decisions of who the organism is based upon as good of a look as you got at it. So this is kind of the first group's Q set. This is the first group's organism right here that they're trying to make that decision on. There's group number 2, group number 3, group number 4. That's one of those difficult to see ones in the field where the bee is just not cooperating. Bee number 5, also slightly difficult. Bee 6, 7, and 8. And so when we were doing more online stuff with students, I had a little kind of like cahoots thing where they could basically say, yes, this is what I'm seeing, no, this is what I'm not seeing. But what I found is that in a classroom, it's just way better to have the student groups talk amongst themselves to figure out if they think what they're seeing is Bombus huntii. And so usually what I'll do is I will leave just this one up on the board, and then I'll have them with their cards as a group decide what they've got. And then the most important part is not necessarily did they get it right, but having them go through and discuss what they're seeing on that bee that's allowing them to make the conclusion that this is huntii or this is not huntii. So we're doing a little bit of CER here, right? A little bit of claim evidence reasoning, but in a very, very light fashion, so much so that half the kids don't even realize that they're doing it. And I wouldn't say CER to them because all of a sudden a lot of them, their brains will turn off because they're like, oh, you've tricked me. And so after they've gone through the huntii, and if we go through this, what we can see is usually I would say like 95% of the groups usually get what is true huntii out of these. So these first three are obviously huntii. This number 4 is a little bit tougher. Other photographs in INAT from the same observation, I can tell this is huntii, but this is one of the ones that causes red flags for students because they're like, I can't see the whole thing. Same thing with number 5, but this is great. You want them to vocalize that. And then basically 6, 7, and 8. 7 is huntii. It just is a really old bee that's lost some of the hairs, where 6 and 8 obviously have different banding patterns. And so getting students to kind of speak to those is really important as far as the CER sort of aspect goes. After we do that round, I say, okay, you guys did a really good job of that. You know, what did you look at on the bees again? Oh, you looked at the banding pattern. Well, let's talk about how people actually came up with these keys over time. So originally, when people were first starting to kind of describe bees and bumblebees, they would draw pictures of them, right? So that way you can communicate what you're seeing to somebody else. And then what you start seeing is they go from kind of these, I would say like nice, almost like Renaissance things with, you know, nice aspect and things like that, to then these more like what we consider like scientific literature looking stuff. Like here's my bee mounted. Basically, they're probably drawing ones from pinned organisms, right? But this allows us to see those key features really easily. And then what we see is in the evolution of guides from bumblebees, somebody then goes, well, if we're really just looking at these banding patterns, let me just derive that down further. Here's all the segments of the bumblebee. Here's the head. Here's where we could have fur on the head. And now we'll just code these out in very kind of geometric looking things, not these fancy photos. In fact, this is traditionally what you'll see now in most Bombus guides is you've got all the different places you could have variation in color and hair gridded out. And then these guides will basically paint in what the differences look like. And so this is traditionally what you'll see like in the National Forest Service guides at this point. So using that kind of guide, what we could do was we could have the students kind of derive a representation of Bombus huntii from what they saw. And so I could have each of the groups kind of paint in their own. And usually what we see is they say, I usually start them with the head. Like, what do we see at the head? It's black, good. Top of the head, yellow. And then the main kind of thorax piece, yellow, with a little bit of a black bar between the wings. And then as we come down the abdomen, yep, we've got yellow, 2 big bars of orange, followed by yellow, followed by black. And so now the students have derived kind of a contemporary description that you would find in a field guide for huntii. And depending on the student group, this can happen relatively fast. If there are a bunch of kids already play games, I've realized like this moves really, really quickly. And then I would then move them on to kind of round 2, which is we're now going to try and look at 2 bumblebees that occur in overlapping counties in Colorado. And student groups will have basically a sheet with their bumblebee on there, and they're just trying to decide which one it is. So we're going to look at Bombus civicola and Bombus centralis. So we've got 2 ways for students to see the differences, right? We've got the kind of classic identification guide now of the gridded geometrical structures. Then we've got a photograph of each. And what you realize is they look kind of similar, right? There's a lot of things that are very, very close about them. The major shift, though, is where that orange double banding is on the abdomen. And then if we look at the distribution of them, They're basically in most of the same counties in Colorado, if we're looking down at the bottom of the slide. And then there is an elevation overlap with them, but Bombus civicola likes to go a lot higher than Bombus centralis does. But they still are doing a lot of overlap, kind of like in this, 8 to 10,000 foot elevation. So we give the students basically these 8 bees to look at, and what they're trying to do is more or less bin them. And so this, depending upon the student groups, this can be really quick or it can be a lot of discussion. And I kind of just let it run for as long as I'm hearing good critiques going on that are staying focused on the subject matter. And really importantly, there's 3 bins here for them to put the different bees into. There's civicola, there's centralis, and there's unsure. And in each one of these, when we talk about this as a group at the end, I want them to be able to do that CER again, kind of the claim of what it is, and unsure is a claim. I am unsure that we can tell what this is, and here's my evidence for not being able to tell. And they do a really great job with this, especially if you've done, of course, round 1, because you've got them used to using that language. So at this point, they should be pretty good at rolling this off their tongue. So we'll have all the groups do that with the bees, and then we'll basically do a big discussion on the sort. And depending on how you run your classroom, you could do this on your whiteboard and just have your 3 bins and just write the numbers next to them. I have people that will just use this PowerPoint and then just put X's on things in order to do that. Or when everybody was on Zoom, we'd literally just have them put their little emojis right on the Zoom screen in the boxes for each thing. And what we find is with this is that there is a lot of these that can end up in the unsure box, right? Partly because you can't see all of the definition, or on some of them, you can see most of it, but maybe you're missing 1 or 2 bands as far as an angle goes. And so there's a fair number of them that end up in unsure. And this is actually specifically done this way. So that way you can say, well, are there other tools that we could deploy here? How could we sort out the unsure pile? And so students will come up with ideas, and usually they're great ones, right? Can we get better photographs of them? Can we get a different look at them? And I like to bring to their attention that there's other tools out there besides just visual identification now that have really kind of accelerated our ability to see species diversity within the last couple decades. I mean, a lot of this goes back to Carl Woese, right? And finding that entire new, basically, branch of life, the archaea, doing this using 16S sequencing way back in the day when you're still using kind of hot DNA in your lab. But now we've got all these tools that are actually relatively accessible, where you could even do it in a classroom. I have tons of folks that just do this in their garages at home now. And so this is becoming a more commonplace tool in order to see diversity. And so I tell them is, instead of giving you new pictures to kind of rebin with, how about I do this? I will give you barcodes of these 8 bees, and let's see if we can figure out who they are now using the barcodes. And remember, just like visual stuff, barcodes aren't 100% all the time. You can have errors that take place in sequencing. They usually happen at the beginning and the end of sequences. So I like to make sure students are aware of that, because if we see that there's variation, say between 2 organisms I'm looking at, and it's happening right at the end or the beginning of a sequence, I would put less kind of confidence that that difference really exists. So there's some really easy tools to do this, and one of them is probably one that you already teach, which is BLAST. And so for image 1 and image 2, these are 2 bees that usually the students will bin as separate. So Bombus civicola and Bombus centralis. And so what we can do is we can take those molecular barcodes and we can just go to BLAST, drop them in BLAST. If you just go to, if you just go to Google and you type in BLAST, then hit go, take that first link, you'll get to your normal BLAST viewer, which we see down here. And then there's a little button down below where you can drop your DNA sequence that says align 2 or more sequences. So what I can do is I can take the sequence from Bombus number 1, I can drop it in this top field, I can take this sequence from Bombus number 2 and then drop it in the bottom field and then just hit go. The QR codes here, the ones on either side of the image, that would get you to the DNA barcode for each of these. You could scan those and play with those if you want right now, or we'll send all these out, of course, when we send out the update for this webinar, so you'll have access to those. So if we hit this into BLAST, what we see is we get this alignment of these two molecular sequences. And if we go to the identities portion of the alignment, we see that they only match up for 93%, which is pretty small in terms of like species. So 613 out of 658, and this is kind of matching the number 2B to the number 1B. And how does this compare to our visual sort? Well, I mean, for some of the groups, this would have been much better because with number 1, they were having a hard time because they didn't have a good look at the upper thorax, or yeah, the upper abdomen, sorry, on that Bombus civicola. So it's just for them to be aware that there are different ways, different tools, right, that we can use to solve the same problem of what is the identity of this organism. And what you could have them do is they could go through now and do a matrix and compare every B to every B. And if you're looking just to have them glaze over in their seats, that is something you can do. Or if they're really good mathematicians, they can set up a great matrix and do this. But this is usually not the way anybody would do it. After you do like a head-to-head comparison of 2, you're like, you understand how that works. And now we can basically put that on steroids by going to something like phylogeny.fr. And just FYI, phylogeny.fr just changed their platform recently. And so when you go there, it'll mirror you out to ng phylogeny.fr, and then there's the tools that I'll be using there. And so now what we could do is, if we're trying to ask the question about how are the 8 Bs that we're looking at in round 2, how do they bin if we look at the molecular signatures, I can basically grab all of those DNA barcodes. And for bees and for most insects, when we talk about a DNA barcode, it tends to be cytochrome oxidase 1, or at least that's the first pass that we usually do in taxonomy for insects, is cytochrome oxidase 1. So what we can do is we can take all 8 of those molecular barcodes and basically drop them into phylogeny.fr in their kind of one-click function, drop them all in there, and then hit submit. And what this will do is we'll take all of them and it'll do that comparison I jokingly said you could have your students do. It's already got an algorithm set up to do that. And then what it'll do is it will give you a binned tree of them, and it will show you how they are related to each other. One thing to be aware of with this bioinformatics, there's something that people call a FASTA file, F-A-S-T-A. And this sounds really fancy. Like you'll go to websites and they'll say, you know, please give me your FASTA file. And you're like, oh crap, do I have to figure out how to make that? All a FASTA file is a text file. And then above your DNA sequence, there's just a greater than sign, and then the title of whatever your organism is or whatever your sequence represents. So the FASTA file that I'm using with the students here literally just says greater than Bombus 1, the cytochrome oxidase sequence, and then greater than Bombus 2, the sequence, and then so on for all 8 of those. Nothing fancy. In this data set, one thing to be aware of, though, I have those 8 Bombus that the students were looking at in round 2, and I have what's known as an out group. An out group is just a way so that if you're doing a phylogenetic tree, if you don't have something that's like not right in the thick of the tree, you'll get a messy tree really easily. And so having an out group basically gives you a trunk to the tree, so that way everything else can align well. And so in this case, I figured we would just choose the honeybee because it's kind of close, but nowhere close to being in the weeds with the Bombus. And so we've got our 8 species of Bombus in this file and Apis mellifera, the honeybee. And so when we run this in phylogeny.fr, what we see is we get a binning like this. And this is really interesting because the students will go, ah, cool. there's 3 bins. There was an unsure pile even for the molecular stuff. And it's like, well, let's dig a little deeper into that first. So let's talk about how these trees display stuff and what this means, first of all. So we've got Apis mellifera at the bottom there. That's our honeybee. That's acting kind of like the quote unquote trunk for our tree, our outgroup. So everything, if we go up from the trunk, is kind of branching out, right? And so we've got 2 definitive lineages splitting out really early on. Bombus 2 and 7 fall into one. And then the top half of that tree, we have another split. We've got Bombus 3, 4 falling in together, and then Bombus 8, 1, 5, and 6 falling in with each other. And if I have students go back and look at their unsure calls from round 2, this doesn't really match up with their unsures. In fact, what they'll see is that a lot of the stuff they are really confident about actually ends up falling into a couple of different of these bins. So Bombus civicola usually ends up as this kind of major branch. Bombus centralis ends up down there visually for them. And then we've got these ones up here, which some of the students may have looked at and called Bombus civicola. So this is really interesting because it's not necessarily a molecular unsure pile. It's a, we're sure these are related, but they're not the same. And so what they could do then is they could go and they could grab the Bombus 1 and the Bombus 3 sequence from that FASTA file, and they could do that blast again, right? And see how close are these. What we see is they are really, really close. So we've got a 99% identity hit. So 653 of the bases are the same out of 660 between Bombus 1 and Bombus 3, which is basically the unknown group and the civicola group. So what the heck is going on here? Are these just errors in the sequence? Well, let's dig into the sequence. I'm going to look at, and usually my rule of thumb is I look at the first 50 bases, and it's like if I see errors in there, I'm like, okay, there's something else going on here. It's not just the sequence. So I'll look in the first kind of 50 here. Yeah, those look pretty good. I'll look at the bottom 50. Well, there is 1 in there, so I have a little bit of distrust. But let's go, but that doesn't account for all 7 of those bases. And so if I start looking through the rest of the sequence, these gaps that you see, this represents where you have nucleotides that are not the same in each of those. And what I notice is I have those spread all throughout the sequence. So this gives me a lot more confidence that those are a real difference and there's something going on here. And in fact, there is definitely something going on here because in April 2021, a really interesting paper came out about a group that was doing exactly this in Colorado. They were studying Bombus civicola, and they were looking at for population dynamics. So they were collecting bees, doing molecular work to try and understand, does a population in this valley make its way over to this valley, et cetera. Because this tells us to some degree, right, how resilient these organisms will be when we talk about climate change, especially if you've got these things that are occurring only at specific elevation band points, because they're going to have to move based upon temperatures and potentially if their forage is moving up or down due to climate change. So in the process of doing this, though, they had this problem where things were binning not into 1 bin, but into 2 with their cytochrome oxidase 1 data. And so what they've proposed is, and this was based upon the molecular information, but also based upon other information that they saw in those environments, i.e. kind of foraging habits and things like that, that there was actually 2 species here, one of them kind of hiding in plain sight. And so they named this one Bombus incognitus, because it was kind of incognito inside of the Bombus civicola group that was there. And if we look like the distribution of where they're at, what you realize is they are happening for the most part in the exact same zones. And so what's maybe taking place is you're seeing a division of species, right, due to some sort of impending factor that we haven't been able to really see. So what 2 things might be driving that? Well, from an evolutionary point of view, right, we usually talk about allopatric speciation or sympatric speciation, i.e. we've got some sort of geographical isolation that's taking place. Remember, this is the front range of the Rockies. There's definitely the potential for allopatric speciation because you've got a lot of up, down, a lot of valleys, rivers, cold spots, things like that. And then you've got the idea of sympatric speciation, where you've got things like reproductive isolation taking place, or maybe the emergence, say, of one at a different season than the other, or maybe even a change in what they're foraging that can cause that split in species. And so this is still unclear with Bombus incognitus as far as I am aware. So I randomly happened to be up in that habitat maybe like 7 years ago in one of the valleys that they'd studied. And so this is one of the valleys they're looking at. And so if you're trying to figure out allopatric versus sympatric speciation, it's not cut and dry, right? Because you've got this elevation, you've got change in forage. And so what I might do is I might give students this photograph and ask them, how would you set up experimentation to figure out what's driving the speciation event? And of course, what I'm hoping for them to get at here is just ways of thinking about the environment that might drive that. Not necessarily anybody's going to derive the answer right away, but ways that we might be able to see that and quantify those differences leading to the speciation. Why is recognizing species really important? Well, if those species, right, are doing things like pollinating our blueberries, pollinating our other crops, and we've got diversity there that we can't see, it means that we might not be setting up appropriate conservation efforts for capturing that diversity that may be helping us, say, make sure a crop is pollinated at a higher percentage than others. And so by recognizing species, it makes us possible for us to conserve them. And remember, all of these things all together build that entire ecosystem, and we rely upon that all the time for our nutrient cycling, ultimately our food, our clean water, our air, everything. So being able to recognize stuff is really important, and those molecular tools are some of the newest tools for us to be able to see that. And just to highlight once again, there's a lot we don't know. 86% of species we still do not have a name for. And a lot of that, yes, is playing in the microbial space, but as you can see from this case study, some of that is still happening at these macroorganism levels. Insects, I think there's still a lot of hidden in plain sight type of stuff going on in there, especially in North America with moths, because moths are really hard to study because they're kind of ephemeral, right? They come out for a couple weeks. And unless you have these really big showy moths, some of these smaller ones, when you talk to experts, I'm like, how do you guys define differences? They're like, well, you've got to dissect the genitalia underneath a microscope. And I can tell you from experience, microscopy after dissection leads to really difficult to call sizes and all kinds of things. And so molecular tools there are making a really, really big difference. I'm an amateur mycologist by kind of passion, and we've been seeing that this is making a huge play in fungal diversity understanding in North America. So I'm in Western North America, where a lot of our macrofungi have been given European names because they kind of, the mushrooms, the fruiting body portion of the fungus looks similar to stuff we saw in Europe. If you do a little microscopy, it might look similar, but maybe not quite, but maybe not enough that you're going to say a new species. But as soon as we do DNA sequencing, it becomes really obvious that you're dealing with another species. In fact, I was working with a group this year called the Fungal Diversity Survey, because they got funding in California the past year to do 5,000 vouchered specimens from across the state. And I would say about 30, maybe 35% of those, when we did sequencing on them, have never been sequenced before and are probably going under the wrong name if they even have a name at this point. And so these molecular tools are really allowing us to see a lot of diversity that's right around us. And just to kind of put it out there, these aren't people like going into pristine habitats. I mean, a lot of these kind of new species are happening like literally around the edge of urban centers. And so remember, there's a lot of diversity hiding in plain sight, and these molecular tools can allow us to see that. The Bio-Rad barcoding kit is a great way to kind of get students doing this. And it's really cool because at the end of this, they've actually generated primary data about an organism in your environment. And if you want, you can have them submit that to GenBank. And now I like to jokingly say they're soft published, because they could say, hey, yeah, I've got data out there in the world. You can go to GenBank. Here's my sequence. So the barcoding kits we have allow you to go after fish in grand detail. I'll swing back to that one. And then there's another kit that comes with a series of primers. You get primers for vertebrates, you get primers for insects, and you get primers for fungi in there. And so you could have students kind of pick from across a diverse group of organisms to look at in your classroom. I really like the latter one just because I'm into fungi and I feel that insects and mushrooms, there's a wild diversity there that still needs to be tamed, and academics really aren't getting pulled in to do this. And so a great place for this data to be generated from is from high schools and colleges as like undergraduate sort of programs. And there's also a big push of this happening in the community science world. Let's swing back and talk about the fish barcoding one just briefly. The fish barcoding one was actually the first kit we put out there in terms of barcoding, mainly because at the time, like 2015, there was all this hoopla about fish at most of your markets not being labeled correctly. And so this was a tool for students to go out, take fish from their favorite sushi restaurant, take it from their local grocer, and basically ask, am I seeing the appropriate fish here? And it still is a horrendous number, like 30 to 40%, especially if you're in some of these coastal states where there's substitution taking place, especially because if you think about fish in markets or at sushi places, right? At this point, you don't have all the morphological features of the fish anymore. All that's been removed. All you've got is a fillet. And so kind of a white fillet of fish, is that tilapia? Is that red snapper? So you put a little sauce on it, it might be hard for some people to tell, but there's been a lot of market substitution in the middlemen space because red snappers, almost twice as expensive as tilapia. So there's this big economic driver that drives a lot of this market substitution, but your students can kind of help find that that's going on and identify what substitutions have taken place. You could also use this in not a CSI food world sort of situation. And you could use this on local fish populations. So I have had some groups that will collect local freshwater fish that are sometimes hard to identify because they're really, really small. And they can basically do clips from them and then figure out what that species is. And there was also a really cool project going on the southeast portion of the United States for a while where they were trying to figure out what invasive lionfish were eating. So when lionfish derbies would take place, people would take the guts from them, pull out the fish inside, and then barcode them. And then we could know exactly what the lionfish were kind of chowing down on at the reefs. And some of the data coming out of that has been really, really interesting because lionfish are very diverse eaters. They will basically go after anyone. But a really kind of cool way to think about using one of these kits. Things that we know that have worked really well are in this chart right here. Insects, beetles, moths, butterflies, bees, wasps, and flies do really well. Spiders are a little bit less robust, and spiders, having worked with a couple of spider experts, there's actually, you need to kind of change your primers a bit and actually look at some different regions in order to get good usable data out of spiders. Things that are difficult or not very robust are molted exoskeletons. There's not a ton of DNA left there. Cocoons also aren't great for the same reason. And gastropods are a little bit tough. Mammals and birds, anything basically with a backbone is pretty easy to get a good sequence off of. Less robust, surprisingly, elk and turkey. I'm not really sure from a scientific point of view what would be driving why those ones are not as great. But things that are difficult to sequence from tend to be ground meats because they've been oxidized and then also nitrite dumped a whole bunch of times, which makes it sometimes hard for PCR to take place. Fur, feather, and bones. Experts can get DNA off of those, but in the classroom environment, they're not really robust. And then for fungi, fresh mushroom caps are really easy to work with, but really the best tissue to work with is gill tissue. So you can work with gill tissue from a fresh mushroom or even dried samples, which is kind of nice because if you're having people do field collections, you can totally decouple when you do the collections and when you do this lab by just dehydrating the mushrooms and then keeping them dry until you go to use them. For fish, most not fatty fish work really, really good. But as soon as you get to these ones that have a really, really good omega-3 content, it starts to not be as fantastic. And that's mainly because a lot of those fats kind of can start interfering with the way that the DNA is cleaned up. And then what happens in PCR is you just don't get as robust of a amplification. Difficult to go after fish tissues, tend to be anything that's been fried, no doubt. Canned fish, because they oftentimes will go through major pressure cooking, which can damage a lot of the DNA. And then sea urchin, red tuna, mussel, and clam don't very frequently in our hands produce good results. From a workflow point of view, it more or less looks like this. You would take your tissue from your organism, you would go through an alkaline lysis system that Bio-Rad's developed that gets you really, really high quality purified DNA through that alkaline lysis applied to a column. And the beauty about this compared to other systems I've worked with is because you're getting better DNA to start with, your PCR reactions are more likely to work. So I've usually seen, a 10, not tenfold, but like, a 10 to 20% increase in the number of PCR reactions that succeed when doing these column chromatography cleanups versus a different method. Once you've got that extracted DNA, they basically just build a PCR reaction, set up that PCR reaction on a thermocycler, let that run, and then run a gel to then see, did I get amplification in the range that I would expect? Most of the primer sets we've built, you're looking for amplification somewhere between like 600 and 850 base pairs. If you see that in your gel, you've got good amplified DNA for the sequencing. And then what you can do is you can just take your PCR sample and then send it out to, there's many companies out there that do Sanger sequencing these PCR samples. And usually within a few days, they can send you the sequence back for you to do your analysis. What I always tell people though is attempt to go talk to your big university before you go to one of those kind of off-the-shelf companies, because a lot of times these universities are running core labs where they'll have a Sanger sequencer, and it's good for you because you're building kind of relationship with an educational partner in your pipeline, and it's also good for you because it's usually a lower price than sending it out to a company. But even when you send it out, it's pretty cheap. Like I've been using MicLab recently in South San Francisco, and I think I'm getting a single direction for about $3 or $4. So sequencing now is very, very cheap as long as you can kind of generate your own PCR samples. And once again, just to kind of highlight why that PCR works so good with the Bio-Rad system, it's mainly because of this lysis system where we're then going to go through and do a cleanup on a column. So those columns are going to use basically a matrix that allows the DNA to bind to it and then allows you to wash out all of the other cellular junk. So you're able to wash that column multiple times to remove all the lipids or as much as you can, proteins, any of these other macromolecules that can sometimes cause problems for PCR reactions. And for fungi especially, you're able to wash out a lot of these pigments. A lot of these pigments can be difficult for PCR to kind of make their way through. And so this process, though, does require the use of a high-speed centrifuge. And so we usually point people to our 16K, so 16,000 RPMs. It's just enough that you can really kind of push those things through the column relatively quick. You can do it in lower powered systems. It just takes a little bit longer. The other big benefit of the Bio-Rad kit is the way that we design the primers for this. So all of our primers are going to amplify a barcode region for that specific taxonomy, but then they've got M13 tails built into them. And all M13 is a vector that most of the sequencing places were using at some point. So every sequencing center you go to pretty much has M13 primers in-house. And this actually makes it super easy then to send your samples out to any place and say, sequence this for me, because you don't have to send primers along, which you would normally have to do in other cases. You can just say, hey, my PCR products have M13 primers, use your in-house M13 primers on them, and they're able to turn that around really, really quickly. So it takes out a couple steps on your end and their end, which also will give you better ultimate data. Like I said, the kit's been out there for a handful of years, since 2015, I think, for the fish. In 2016, I started kind of playing around with the fungi stuff with some local schools. And it was really fun because I find this is also a great community builder. So this school ended up working with their local water municipality and said, hey, we've got this cool tool we're going to be using at the school. Would you like help identifying the organisms in your watershed? Because in a lot of these water municipalities, part of their charter is also to understand the organisms in the watershed. And some organisms for those agencies can be very tough, like fungi, like moths. And so this group went out and basically did a collection of the fungi from the watershed, did barcoding, and then provided the data back to them. And it was really cool because the agency got really excited about it because all of a sudden they're able to see and identify new organisms in their area. Students got excited because their data really meant something to somebody. And then what I've seen is the agencies come in now and done kind of like more in-depth talks about what it's like to work for the watershed agency, what threats they have to, water safety in the area, all kinds of stuff. And it's kind of created this multi-year kind of community dialogue, but it mainly started because the school all of a sudden had this tool for identifying organisms. And then also within that, they identified a bunch of like new to science species, i.e. ones that had DNA barcodes were radically different than anything that's in GenBank at this point. These haven't been written up into a new species yet because that is actually a lot of work. Somebody needs to go through, sequence everything that's close to this organism, and then do microscopy and kind of field note comparisons to then see, is this something that we can call new, and then move forward. So that's more of a master's level project for somebody maybe from this school. We've also done this in Arizona before, surveying the Chiricahua Mountains with a bunch of local teachers, and have just found a huge diversity of undescribed species down there. But the big take home here is that you can kind of make your own discoveries with these barcoding kits. And like I said, I really like the barcoding kit where you've got the insects, the vertebrates, and the fungi primers, but we also have that fish barcoding one if you're just more of a ichthyologist type of person and want to look at fish or local market substitution. And then, of course, if you've got any further questions, you can always email us at bioradexplorer.com. My name is Damon. Like I said before, I'm on the kind of the West Coast. Tamika and Lee are spread out across the country, and I think Aaron might be out here in a little bit. Did anybody have any questions I could handle? And sorry, I wasn't able to monitor the chat while I was talking, just the way the screen shows. Was there anything that came up in the chat, Lee, that needs to be
I don't think so.
covered?
So we were just talking about the sequencing costs and whether or not that included cleanup. Usually they do include like a generic cleanup, it seems like.
Yep.
I will add, I just looked up UT Austin for a teacher that's local to me. I'm in, just south of there, and they had sequencing for I think $4 a couple of months ago, and I just saw that they suspended their Sanger sequencing service. So make sure that they have that. That's one of the key things. So yeah, but I think hitting up the universities and community colleges is as a first resort is the way to go, and then some of the resources that Damon mentioned.
And if you're trying to find those, you can always email us too, and we can kind of look around and ask people that we've been working with in your area and say, okay, this one has worked in the past. And then if not, like I said, there's a handful of like kind of national providers that do this. So MICLab, GeneWiz, Eurofins, I think is still doing Sanger as well. And you literally, it feels weird just to like pop PCR tubes in an envelope and just send them through the mail, but it works even at room temperature, as long as it's not one you've been sticking a pipette tip in a whole bunch and getting contaminants in there. Remember, those PCR products are sterile, so they do preserve well.
Nikki said that they had great looking DNA on their gel, but the sequencing failed. Any suggestions? That was what GeneWiz with M13 primers.
Ooh, I would come back to GeneWiz really quick and be like, here's my gel. Can you guys run that again? And a lot of times these companies will. I had GeneWiz or AstraZeneca or whatever they are now, I think, fail on me about 7 months ago. And I came back to them and I was like, here's my gel. Can you guys run this again? And they ran it again, and I actually got sequenced back. So and these companies a lot of times won't immediately come to you and be like, oh, hey, can we run this again for you? So you kind of need to push them a little bit and be like, hey, can you guys run this for me? I think this might be on your side.
Eric wants to know if we have a good workflow, license free for reading and base calling Sanger sequences once they come back.
So people use a variety of things. I personally use something Google developed years ago when they thought they were getting into the genomics world called SeqTrace. And what I like about SeqTrace, and I'll just type it out in the chat, is that it is very, very minimal. So all it allows you to do is basically see your chromatograms and then take them and try and make contigs. So it's like literally just perfect for this type of barcoding stuff where you might be doing a forward reverse read, making a contig from that, and then using that. There's a lot of other kind of like free ones out there, but SeqTrace I find when using it with the students, there's just not a ton of bells and whistles, which is perfect.
If you really want to get into the whole barcode of life thing, there is a student, well, it's a one that you can't submit sequences to, but that you can use it for, and it'll do that as well. It's a little bit convoluted to use, but it does okay with that. Let's see, any other questions from anyone? I put the survey in the chat. If y'all don't mind taking that, that would be great. Just mainly to get your feedback on what other webinars you'd like to see, or if we need to answer any questions, you can put them in there as well. And I'll also add on our link for our other webinars. We don't have our current webinar list up. It will be published within the next month, maybe sooner. So check that out. We'll start adding on things from May through August pretty shortly in the next few weeks or so.
And if anybody ever needs more in-depth information on sequencing, bioinformatics, and things like that, feel free to come back and ask. Like I used to work very deeply in the genomics world. I worked on the first draft of the Human Genome Project, and I've worked on a couple of the sequencing technologies like since then. So I at least can, if I don't have the answer myself, I can at least probably point you to the right person that would have it.
Okay. Well, if there's not any more questions, I'm going to turn off the recording. I'll send this to everyone, and we'll send the PowerPoint as well. And if there's anything that we can do, like I said, feel free to reply to me and I'll get you to the right spot. Thanks so much for coming.